So I would like to introduce the second speaker, Robyn Meurant. She came here from NSF, the Public Health and Safety Organization. And Robyn is the Executive Director for Regulatory Consulting with a focus on IVDs and EU regulations. She has more than 30 years of experience in the field of IVDs and has served as senior scientist in large diagnostic laboratories, being the regulator of IVDs with the Australian Therapeutic Goods Administration. And before she joined NSF, she has been a senior technical officer for pre-qualification of IVDs at the World Health Organization, being in charge of the technical file assessment. And today she'll focus on the extent of clinical evidence required for conformity assessment. Thank you very much, Robyn. The stage is yours.
Thank you, Sandra and BIOPRO, for the invitation to talk, and for Michael for proposing me. It's great to be here today, and also to be talking with my ex-WHO colleagues, because at WHO I worked with Michael and Misha Nubling and with Sabina Ross. So it's great to be all back together again. And today I'm going to talk on the extent of clinical evidence required for conformity assessment. So we're introduced to the topic of clinical evidence for the first time in the regulation in the recitals, and the recitals are the why part of the regulation. And it tells us that to ensure a high level of safety and performance, demonstration of compliance with the general safety and performance requirements laid down in the regulation shall be based on clinical evidence. So here's our first introduction to the concept. I'm really struggling here, sorry, with the screen. So clinical evidence is made up of data generated to support the scientific validity, the analytical, and the clinical performance. Its purpose is to demonstrate the clinical benefits of your device. And I'm sure all of you are familiar with these concepts, but let's just quickly review them. When we talk about scientific validity, it's a term I don't like at all, because everything should be scientifically valid that we do. But in the regulation, it refers to the association between the analyte or the marker of your device and a clinical condition. So then when we talk about analytical performance, it's about how good your IVD is at detecting the analyte or your marker or markers. But clinical performance is about how good your IVD is at providing information correlated with the patient's clinical condition. And you need evidence for all of these, and that's your clinical evidence. The clinical data and the performance evaluation results comprise your clinical evidence. And you're expected to demonstrate clinical evidence for all IVDs unless any omission can be justified as not applicable. So the answer to our question today of how much clinical evidence we need is simple. You need a sufficient amount of evidence that is of sufficient quality to allow a qualified assessment of whether the device is safe and achieves the intended clinical benefits when used as intended. Well, that sounds simple, doesn't it? Anyway, let's go to this little graph, and I hope you can see that well enough. There are many sources of clinical evidence, and generally most of our evidence is generated from studies that we perform, but there can be other sources. And here for scientific validity, we often refer to academic research when we're looking at the clinical association. We can see the clinical literature, feasibility studies, clinical trials, clinical performance studies, and consensus expert opinions and positions. I'm not going to read all of these, but this is to give you an idea. When we talk about analytical performance, we often talk about analytical sensitivity and specificity and issues like that. For clinical performance, it's a shift because we want to see how well the device is in being useful for the clinical condition, and there's a number of different means to show that. There's a number of different types of tests or different performance characteristics that we may look for. Just wait for my slides, they're taking a long time to come to you, sorry. Here we see the intended purpose, as Sabine very, very clearly pointed out to us, being a critical aspect. And it dictates the type of clinical evidence that must be generated and therefore drives the process that ultimately results in sufficient evidence to demonstrate the clinical benefit. So we know we need clinical evidence to meet the GSPRs, the General Safety and Performance Requirements, and to demonstrate clinical benefit. But how much is that? Is it a little? Or is it a lot? So I want to show you here that different IVDs will need different amounts of clinical evidence. And before we dive into the topic, let us look carefully at this diagram. IVDZ on the right-hand side has significantly more clinical evidence than IVDX on the left-hand side. Yet for all three of these hypothetical IVDs, there's roughly the same amount of analytical performance required. And this is generally true. There are exceptions to this rule, such as some Class A devices and IVD software where analytical performance may not be applicable. We will discuss some other exceptions to this rule later on. Another point to remember is that the regulations allows that in justified circumstances, clinical performance can be demonstrated through means other than clinical performance studies. Other avenues include use of scientific peer-reviewed literature and published experience gained by routine testing. There is a caveat to this in that we are informed in the regulation, though, that if testing in an end-user setting is omitted, that it should be justified that the internal conditions of use, for instance, your R&D laboratory, that they cover the normal conditions of use as described in your intended use statement and in Annex 1. So let's start looking at some aspects that impact on the amount of clinical evidence required. Remember, every IVD is unique based on its intended purpose and its manufacture, and all the following aspects that I'm about to discuss must be considered when judging how much evidence you will need. Firstly, the class can dictate, and normally the lower class, and I'm sorry you can't see the bottom of my slide here, I just have to shift it up. The lower the class, the less clinical evidence. And why is that so? Because our classes are about the criticality of the result to the patient and to public health. They're based, the risk classification rules are based on the impact to a patient and to public health. So the higher the risk class, therefore it's reasonable that the greater the level of clinical evidence you will need. So now let's examine in more depth the device and its intended use. What is known about the marker you have chosen or the technology you have used? Where the marker is novel, for instance, and I'm going to give a hypothetical device. A new biomarker has been discovered and we believe it's useful for monitoring of cancer treatments. So in this case, compared to an established device, they're both going to be Class C because it's a genetic test. The new biomarker needs to have both scientific validity established and the clinical performance. So the scientific validity question, the evidence you need to generate for this is, what is the association of the marker with the treatment of cancer? And the evidence we need to supply for the clinical performance is, can we show that the assay successfully monitors the progression or regression of cancer in accordance with the intended use in a manner that is useful for a useful tool for clinicians? So to do this, we may need to do proof of concept studies to establish scientific validity, or we may incorporate all these into a study that will answer a lot of the question around scientific validity plus clinical performance. And now what if we have a novel technology? Here it's possible that we're going to need a lot more evidence around analytical performance because we don't have a proven track record for this new technology. So we're going to have to provide much more evidence about the robustness of the methodology of the new technology, around its sensitivity, its precision, and aspects like this. And it may also therefore impact on the amount of clinical performance data we're going to need to provide also. Now let's look at the intended purpose, the critical part of everything, as Sabine mentioned. So the function of the IVD, whether it's for screening, for diagnosis, for use as an aid to diagnosis, will dictate the type of studies needed and the level of evidence required. An aid to diagnosis may not need the same assurance of high performance qualities as an equivalent device just for that is required for diagnosis. Now I've taken this slide. This slide is from the MedTech Europe recent clinical evidence requirements document, which is actually excellent, I have to say. And what was great, it's only recently been published, but it confirmed all my thoughts and the advice I've been giving on clinical evidence and levels. But here we see it talks about the function of the device. And on the left-hand side, we have the intended purpose: screening, diagnosis, disease monitoring, etc., etc. But when we look at the study design on the column second from the right, I hope you can all see that and read that well enough, but it talks about the type of studies, and you'll see that quite often it'll say prospective or retrospective, but that's not always a choice. So there will be some devices will need studies that only need to use leftover specimens. And this translates into considerably less effort and less evidence quite often than is needed for assays that may require a prospective randomized control trial. And perhaps a good example of that is the amount of clinical evidence we may need for a genetic test that just identifies different genetic mutations that can be found in certain cancers, versus the evidence we may need for a device that detects the same genetic mutants but is used as a companion diagnostic. We certainly would be expecting to see prospective and randomized control trials for the companion diagnostic. And similarly, this diagram illustrates the difference in requirements for an assay with the same marker but different intended use, depending on the criticality of the results to the welfare of the patient or the impact on public health. So another example of this might be a test for screening of the blood supply, where wrong results can be catastrophic for many potential recipients. And so therefore, it might be, let's say, a syphilis test, which can have just an intended use for diagnosis of syphilis, or it may be for screening of the blood supply for syphilis. If it is for screening of the blood supply for syphilis, we're going to need a lot more clinical evidence to have the assurance that public health is looked after. So what else do we need to consider? We need to think about the context in which the device is used, who the users are, why are they using it, what are the potential sources of error. The IVDR, I'm sure you all know, has a new GSPR, it's number 19, relating to self-testing and near-patient testing. Regardless of the class, the notified body review of a self-test or a near-patient test includes the review of clinical evidence generated from studies specifically carried out with the intended users. So it is not sufficient to only add studies generated in your R&D lab. The type of studies you may be required to undertake for a self-test or near-patient test includes assessment of the ability of the intended use population to comprehend key messages from packaging and labeling. Do they understand the key warnings, limitations, and restrictions? Do they know how to correctly perform the test procedure from the information provided, or do they need extra training? Some other aspects you may need to investigate, which will create more clinical evidence, is can they interpret results accurately? And you also have to think, have you really tested a sufficient range of potential end users? So let's just wrap up with on the sort of factors that can impact and that you may or may not have thought about that can contribute to clinical evidence. Is there variability in your subject population? And this may be, for instance, I'm an infectious diseases person, so I always think of the different, there could be mutations in certain populations, there could be interfering substances in certain populations. If you know about this, and you should have done a comprehensive risk assessment to find this out and literature review, then you need to be examining those populations, and certainly the level of clinical evidence will increase. Now, just to, we've talked about some factors that impact, but an important part about the level of clinical evidence we need is to demonstrate state of the art, because we're told that for clinical evidence, it shall be such as to scientifically demonstrate by reference to state of the art in medicine that the intended clinical benefits will be achieved and are safe. I have the reference there, sorry. Does anyone know how many times state of the art is mentioned in the IVDR? If anyone wants to have a guess, please go for it. It's actually 20 times. It's 12 compared to the it's mentioned 12 times in the MDR and 5 in the IVDD, but it's not defined. Fortunately, it is defined in the latest version of ISO 14971, which describes to us that the state of the art embodies what is currently and generally accepted as good practice in technology and medicine. It does not imply the most technologically advanced solution, and it's generally described, it can be referred to as the generally acknowledged state of the art, generally acknowledged. But how do we demonstrate state of the art? And standards are one way we usually do. And when we think of demonstrating state of the art for technology, the latest version of an ISO standard is often used. However, at the moment, we have a conundrum because at this point of time, no standards have been harmonized for the IVDR. In addition, many of the useful standards for generation of clinical evidence for IVDR, for the IVDs, are CLSI standards, and not so many. I mean, there are certainly many ISO standards, but there's many useful CLSI standards, and these traditionally have not been included in the list of harmonized standards in the EU. However, this does not mean you cannot use the standards. It does mean, though, that you should supply a justification for the use of the chosen standard as to why it should be used to establish state of the art. And my other advice regarding this issue at the moment, because it is, we are in a conundrum, is make sure you talk to your notified body and see how they intend to tackle this issue. So I'm just up to my final, almost final slide. It's how we determine state of the art in medicine. There are some points which may impact on the level of evidence you currently hold if you have not reviewed your data in light of the new requirements. State of the art of medicine is also not defined, but usually is accepted to mean the current standard of care, of testing, or the current medical practice. Issues to consider that you may not have thought of is that you need to consider state of the art of medicine in the EU. Not what's appropriate elsewhere, but in the EU. This is IVDR, this is CE marking. You cannot assume it's the same everywhere. In addition, beware if your evidence is built for the US and a 510K submission. The predicate device that you use may not be state of the art in the EU. You may need to generate some new evidence. So that's it, is it? As I've described, there is a basic principle normally applied to the level of evidence required, and that's regarding the class. But as I hope I've explained, a comprehensive risk assessment will identify the evidence you need, and class is far from the sole determining factor. But guess what? It never ends. Remember, the IVDR is built on the principles of continual review and assessment of risk and of clinical benefit. So yes, it never ends. Thank you for your attention, and I would love to talk to you more if you have any questions regarding this presentation. Thank you very much.
Thank you very much, Robyn. Right now, there is a little discussion ongoing in the chat, but I can't see specific questions.
Robyn,
yes,
I'm I'm taking the occasion to ask a question related to harmonized standards in the field of IVD, and I mean it's more a general discussion. We all are aware that the harmonization request has been refused, and I saw the refusal letter. The argument is it is like hitting the face of Commission. What came back for this standardization request, and I think especially in the field of IVD, there is a problem with this harmonization of standards, because as we know that this process is taking quite a long time. I think the state of the art should be referenced by the latest standard, and wherever this standard may come from. I mean, it's science that should count, and not what the standardization body in the end wrote in a standard that may occasionally be harmonized. No.
Michael, I would agree with you 100%. And I wanted to raise this issue because of the two facts, exactly those two facts. Number one, that we don't have any harmonized standards, but regardless, I think everything we do should be scientifically valid and should be, and wherever that state of the art can be established, you should do it. And as I say, I think the safest way to go ahead is use what you know is the best and most valid source of reference, whatever it is, be it a CLSI standard, any document that you think, but provide justification around why you feel that is is for you state of the art, or provides state of the art for your justification, for your clinical evidence. So I think whatever we do, certainly throughout the whole of the new regulation, I think we have to. There's been a big shift in the regulation to me, in that you not just have evidence, but you have evidence that's much more meaningful, that really drives, shows the clinical benefit. So whatever you do, whatever evidence you have it justified to why you can conclude that the benefit outweighs the risk, and there is clinical benefit in your device.
And I think the WHO has shown by example in the pre-qualification program that this is possible, to check on the latest state on the state of the art in science for that, without any need for harmonizing standards related to that.
That's right. WHO pre-qualification has a list of many, and I can share that I'll share the link later on. There's some guidance which gives a list of many useful documents that can point to state of the art. So I'll find the link and share that list, that document for everyone soon. I certainly give that to all of my clients.
So there are two more questions in the chat. One is, do the clinical studies have to be performed externally by an independent laboratory?
No, there's nowhere does it say it must or must not. What you need to do is make sure you have evidence that all your studies are as free from bias as possible, and that independent studies certainly add a level of confidence that others can't. But if you provide independent studies, make sure with your documentation that you can get hold, where possible, of the full protocol, so that the level of every study has a level of bias. But by providing a comprehensive if you can, for any of your studies, have a comprehensive protocol, so that a notified body assessor can see where there may be bias in that study, and how much confidence they can at the end of the day put into the results that you have produced. For me, it's useful. There's pros and cons on both ways, but certainly, I mean, an external study will also show that your instructions for use are appropriate, that the results and the test can be performed acceptably. But again, the assessor will need to use that evidence. You will need to be able to show that the manufacturer, did not interfere with the testing or do anything other than provide the amount of training you state. So I think you can do both. but happy to have maybe Sabine come in on that conversation if she's available to see what she's expecting as a notified body.
Yeah, yes, I am available, and I just entered something into the chat. It's really complicated because there is no right or wrong answer here. As Robyn mentioned, it highly depends on the knowledge of the product, the knowledge of the group of patients, the knowledge of predictable values. So you should know a lot about the parameter and also your product, how it works, where it needs to be used, and who would use it on whom. And then you have to establish the knowledge about where potential patients can be found. It's not that easy to find patient samples nowadays, but in most countries you will find excellent centers, and you might approach those, or key opinion leaders, you might approach those clinical laboratories to for help, and I'm pretty sure they are happy to help here. Also, I would like to mention that, of course, if there is data available in the public domain, which is scientific literature, peer reviewed, then you can take that into account to reduce the amount of clinical performance data by justifying that your product has either been used in a review, a scientific review, or is sufficiently similar to one that was reviewed in or tested in a published review or a scientific paper. There is excellent guidance, and I put that into the chat in ISO 29116. If you look into that, you will find a really clear instruction how to plan, how to perform, and then how to assess the results of those investigations. You might want to have a look into the regulation, because it requires the manufacturer to appropriately plan the collection of sufficient data to provide evidence of the performance and safety of the product. So this sounds like really very sophisticated, but in fact my knowledge of industry and looking at technical files of high-risk products is that most manufacturers know exactly what their product does, where and how it is used, and what the risks are. And also the statistic approach to provide evidence of reliability of that data might be considered. So please refer to statistical methods to ensure that the reliability of your data can be agreed on. I think that's really important. It's not just the risk, it's also the reliability and the statistical relevance of the data. We see a lot of manufacturers where they say, "Oh, we use square root 100 or square root of n," or something like this, or 100 is always a good number for sample. You have to just justify why exactly you selected this patient sample number or this number of patients in a specific group. And it is essential that you have an appropriate plan. And if you have already a notified body, please discuss with them if that plan would be agreeable by the notified body, so that you don't work into the wrong direction.
I fully agree. We often see that the biostats is lacking simply science. I mean, there is you can say from literature compared to the claims, then looking to the protocols that are made, that there is no link related to sound biostatistics behind. And that's a big problem. That's also a problem for many manufacturers that today make the claim, yeah, we have sufficient evidence, we have a lot of data. Look into the data in detail and look whether the biostats that are behind there is really sound enough to support the claim that you are making. I think there's another question from Diana Ramirez.
Yes. Is there any kind of algorithm that combines all mentioned variables to define how much clinical evidence is needed?
I don't know of any, and I think maybe I think there are just way too many variables to do this, and every marker is different. We look at, for instance, again, I'll go back to my life as an infectious diseases person. If you have an antigen on a plate, it may detect maybe an antigen against many parts of the virus or whatever, and so the complexity of your marker of everything will impact on that, and so it's just something you have to do. Sit down and you start with your intended use statement, really tease that apart, make sure you know what your intended clinical benefit should be, and do your risk assessment against that, and start collecting your evidence. Starting from the literature, doing a comprehensive literature search is often one of the best ways, and taking it from there. But no, there is no tool, and I think it wouldn't be useful, because at the end of the day, we just need to make sure that we have the evidence and sit down and do it. And again, I think if you have something that needs to go to a notified body, always, I think pre-discussions with your notified bodies are a great way to go, or using an experienced consultant like those at NSF and Medidee and others. That's why we're here, to help you sort through some of this and really get scientifically valid evidence, because you can have dossiers that are thousands of pages big, and they can be full of nothing. So yeah, just make sure anything you do, be it small, be it large, make sure it's scientifically valid and really demonstrates clinical benefit.
Thank you very much, Robyn.